Welcome back, everyone, and welcome to our second session, Searching for Immunity. As we heard in the previous talk, the last almost 2 years have been a struggle, but also an opportunity for the medical fields facing this pandemic, and even more so for the field of research. It is interesting to think that while the healthcare system was battling to offer the best care for those affected by the virus, researchers in the labs were restlessly working to offer a cure and answers to the world waiting. Thanks to the knowledges that has been achieved on the course of the years and the technologies we have today, probably more than ever, vaccine research has been a real-time partner in battling this fight. At the same time, the research field in medicine has been shaken by new questions about the importance of rigor in research, when many research work done in a state of emergency were shared with the scientific community only after preliminary evaluation due to the rush of times. We are very pleased to have here with us today Dr. Mark Painter, PhD and researcher in immunology, who has been working in the first line for the understanding of the biological implication of the SARS-CoV-2 vaccine and the immune response. Dr. Painter graduated from the University of St. Thomas in St. Paul, Minnesota in 2014 with a bachelor degree in biology. He subsequently entered the graduate program in immunology at the University of Michigan. During graduate school, he studied how HIV evades the immune response and establishes a persistent infection. And he discovered a new class of drug that could help the immune system recognize and kill HIV-infected cells. As he completed his PhD in immunology in November 2020, and one of the peak of the pandemic, he began a postdoctoral research fellowship at the University of Pennsylvania in January 2021, which is one of the places where the first pioneering works in the mRNA vaccine field were first accomplished, as it probably will tell us. During this time, he has been working as a co-leader of a team that published several high-profile studies on the immune response to SARS-CoV-2 vaccination and infection that have been publishing in high-impact scientific journals such as Science Immunology, Immunity, Nature Medicine, and Science. So thank you, Mark, for being here with us today. We would like to ask you to guide us through the fascinating journey of the development of this novel vaccine that actually started probably many years ago, and to discuss about your experience working as a researcher in this time of emergency. So thank you, Mark.
Great. Thank you, Giulietta. I will try to share my screen so we can go through some slides. Do you see it? And
yes.
It's in presenter mode. All good?
All good. Perfect.
All right, great. So it's really a pleasure to be here. I'm excited, and actually it was a lot of fun for me to prepare for this presentation and think back a little bit on my experience of the last year. So I'm grateful for the opportunity, and hopefully it'll be interesting to everyone else. I think this is some of the most interesting stuff in the world, personally, so hopefully I can share some of it. So we're going to talk about searching for immunity, the long road to a vaccine, and what COVID-19 is teaching us about biomedical science. And we'll see if I can advance the slide. Yes. All right. So I know we're all medical professionals, and so a little bit of this might be redundant, but I want to make sure we're starting on the same playing field. And I think it's helpful to be reminded of just how some of these things work. So I'm going to start with a brief overview of just how biology works in general and how the immune system gets activated in response to an infection or a vaccine, and then talk about the development of the mRNA vaccines in particular, and then a bit about my experience and what we've learned about science during this pandemic. So in a big picture view, the flow of information inside of a cell, right? So all life forms are made up of cells, and within those cells, this flow of information is really highly conserved. So every life form takes advantage of this strategy, which is that you have information encoded in DNA, which can then be transcribed into messenger RNA molecules that can then be translated into proteins. And proteins are what actually do things in the cell for the most part. This is a lot of what I say will be overly simplified, but hopefully still reflect the truth. So you can think of proteins as the things that actually do things for the cell, that help your cell achieve the functions it has to do in order for life to happen. But the instructions for building those proteins are encoded in the DNA. So I like to think with an analogy, right? The DNA is inside the nucleus. The nucleus is a hard place to get to. It's highly protected, and that's because the DNA is very precious. So the DNA is kind of like your original set of instructions, maybe like the original architect's drawing for how to construct a cathedral. Or a carpenter's drawing of how to build something much simpler like a chair, right? And the original copy, there's only one of them and it's very precious, is protected in the nucleus, kind of like a high security library, right? If you have the right access, you can go in and make photocopies of those plans, the instructions for how to build a cathedral or a chair. And those instructions are the messenger RNA molecule or the mRNA. And the mRNA can then exit the nucleus and go out into the cytoplasm, which is a less secure place. A lot happens in the cytoplasm. It's more dangerous out there. So you wouldn't want your precious DNA there, but since you can make a ton of photocopies, these are cheap, they're disposable, and they carry the instructions that are encoded in the DNA out into the cytoplasm. And then out in the cytoplasm, you have this organelle called the ribosome, and the ribosome is a builder. And it basically will take any instructions it receives and follow them and build, either build cathedrals or build chairs or whatever the instructions are, right? So you have this high value information in the DNA. You make these lower value disposable copies as mRNA, and then you have these builders that kind of mindlessly will just turn whatever instructions you give them into proteins, and those proteins can actually do things for the cell. So this is how our information flows, right? Now, viruses take advantage of that. So this is a schematic of a cell infected with SARS-CoV-2, which is the virus that causes COVID-19, as we're all familiar, I'm sure. So here's a little cartoon of a SARS-CoV-2 virus. It's kind of cute, I think. And it has the spike proteins all on the outside, and it has the viral genomic RNA on the inside. And so what it does is it, through interactions with a receptor, it manages to enter the cell. And as I said, it's hard to get into the nucleus, but since SARS-CoV-2 is an RNA virus, it doesn't need to. And so it deposits its RNA genome into the cytoplasm. And so as soon as this RNA arrives in the cytoplasm, your builders, your ribosomes, perceive it as instructions, and they start to build whatever it tells them to build. So this is how a virus hijacks your cell, right? That your cell is trying to do what it needs to do, build the proteins it needs to execute its functions, in this case, to be a good lung epithelial cell. But when the virus arrives, the instructions encoded on its RNA are read and built, and the cell becomes a factory for all of these virus proteins. These virus proteins can then assemble into new viral particles and exit the cell to go infect other cells nearby, or to leave your body and go infect someone else, right? And so in this way, the virus is able to replicate, hijack what the cell was doing, take advantage of the fact that those instructions are kind of read no matter what they say, whether they help you or hurt you, to produce proteins, and use that to make new viruses. So obviously this is bad, and we've had millions of years of evolution with viruses to learn to counteract this and to protect ourselves against these viruses. And so I think there's really two different levels of the immune response against a virus to try to prevent this from happening. And they're both going to be really important for this talk. So the first is called innate immunity. And what innate immunity is it doesn't have any memory, and it's less specific for any given virus, but it works on the principle that in general, viruses and bacteria look different from ourselves. And so inside every cell in your body, there are these receptors, these proteins that recognize things that look unfamiliar, that don't look like part of a normal human cell. So for instance, our DNA stays in the nucleus. It doesn't go out into the cytoplasm. A lot of viruses are DNA viruses. And so they'll enter the cell and there will be DNA present in the cytoplasm. We know that DNA isn't supposed to be in the cytoplasm. And so if there's any DNA recognized in the cytoplasm, it will trigger this innate antiviral immune response that will recruit immune cells to the area to try to fight off the infection. And will also do some really important things, right? So once this cell becomes infected, it becomes a factory for virus, right? Normally, we want our ribosomes building whatever instructions we provide them. But if there's a virus inside the cell, you don't want that to be happening. And so part of that innate antiviral response is also to shut down the production of these proteins to make sure that the virus can't continue to replicate and spread. And so we have this innate immune response that can recognize foreign material. And a lot of it is foreign DNA and RNA that might come in the form of a virus, right? That's great, but it's usually not enough to actually fight off a viral infection on its own, although it does help limit the pathogenicity of the virus. And so what we really need is an adaptive immune response, which is the part of immunity that has memory and that's targeted by vaccines. And so what an adaptive immune response is that certain specialized immune cells called B cells and T cells can recognize specifically individual aspects of that particular virus. So in the case of SARS-CoV-2, they could recognize this spike protein, for example, and learn to fight off that virus very efficiently. So here is just going to be a quick overview of how the adaptive immune system works, and then we'll get into the mRNA vaccines. So in order to have immunity against a virus, you need to activate these B cells. B cells produce antibodies, which can help prevent you ever getting infected and neutralize the virus before it gets into a cell, and T cells. And you have helper T cells that sort of coordinate the whole immune response, and killer T cells that actually eliminate the infection by killing infected cells. And this, I think, is like really the coolest thing in the whole world, which is why I'm an immunologist, which is that these B cells and T cells, when you're born, your body generates millions of B and T cells, and each one pseudo-randomly, mostly randomly, recognizes one thing, just one thing. And you have millions of them, and they all recognize one thing, each one different from the other. These are randomly generated, and probably most of them don't recognize anything that exists or has ever existed because they're randomly made. But you make them and they go and they wait, right? And if they recognize, if the thing that they recognize arrives, they can respond. So you make this repertoire of millions of B cells, millions of T cells that might or might not recognize something. And I think that this is really fascinating because when you're born, a baby doesn't know what it's going to be exposed to, doesn't know what viruses and what bacteria will try to invade their body during their life, but they generate B and T cells that could recognize anything that will ever exist, not just the viruses and bacteria that have been infecting us for generations, which I think is really amazing because if you think about it, right, human beings for the last 10,000 years have had B cells and T cells that recognize SARS-CoV-2 virus, and they were useless that whole time, but they were there. Everybody has always had them. And these cells that were useless for 10,000 years or more, like suddenly last year became very useful. And you can see the beauty of this approach. But when those cells are generated, they can't actually do anything yet. And that's because you don't want a bunch of inflammatory response against things that don't even exist, right? And things that aren't actually dangerous to you. So when those cells are first generated, we call them naive. So you have naive B cells and naive T cells. If the virus actually arrives, so let's say this B cell recognizes the spike protein of coronavirus, that naive cell can become activated. It undergoes a transformation into this cell we call a plasmablast, which is not super important, that creates a bunch of antibodies. So this B cell will create a bunch of antibodies that recognize that thing that it's specific for. So if this B cell is specific for the SARS-CoV-2 spike protein, it was waiting there your whole life, it thought it was useless, suddenly it becomes useful because the spike protein arrives on the scene in your body, and it will start to produce antibodies that will recognize that spike protein and neutralize these viruses to prevent infection for as long as the antibody persists in your blood. But B cells can also take a different trajectory. they can interact with some of these helper T cells and become a couple of different, a long-lived plasma cell that will keep producing antibody, or a memory B cell, which can actually undergo these mutations, which is really cool to recognize even new variant strains of related viruses, and be able to rapidly produce antibodies in the future. And these helper T cells can become sort of generic antiviral helper T cells. And these killer T cells that were naive, they couldn't actually kill anything. They had no functions. Once they experience their target, the spike protein in this case, they can become these effector T cells that can actually go out and kill the infected cells to help you eliminate the infection. The problem with this, it's a beautiful thing. It's a great thing. And after the infection is cleared, they transition into these memory B and T cells. So the problem is that naive B and T cells take a long time to get activated. They're naive. They don't know how to do anything. So the B cells don't know how to produce antibodies. The T cells don't have a license to kill. And so it takes 7 days, 10 days, 14 days, depending on the person and the virus, for these cells to actually get activated. And so if you think about the context of an infection, that's 7 days, 10 days, 2 whole weeks where the virus is replicating in your lungs relatively unchecked, with almost nothing counteracting it besides those innate antiviral responses. And so this is really like where the drama plays out with a brand new virus like SARS-CoV-2. No, we all have these naive cells, but none of them have been activated before because it was a brand new virus. And so when someone gets infected, it takes 2 weeks to really generate a good response against that virus. And during those 2 weeks, if the virus establishes itself too much, it may be too late. And that person could ultimately succumb or experience a much more severe infection than was necessary because the virus was able to spread so much within their body during that week or 2 weeks while these naive cells were being activated. And during that time, they can spread the virus to other people. And that is really like what drove the pandemic, is the fact that nobody had any pre-existing immunity to this virus because it was brand new. The amazing thing is that once these naive cells have received their license to kill or learned how to make antibodies or become memory cells, they're much more effective. So they're not naive anymore. They've matured and they become these memory cells, and they multiply, and you have far more of them than you had of the naive cells at the beginning before you were infected. And if you were to get infected again, if your antibodies decrease and a new cell becomes infected within your body, these memory B cells and these memory T cells respond rapidly. So instead of 7 days, 10 days, 2 weeks, we're talking a couple of days to be producing new antibodies that can recognize different variants of the virus, and killer T cells that can go help eliminate that infection. And so this is what, these are the different layers of immunity that infection or a vaccine can give you, is that once these cells, these naive cells, have seen that protein, the spike protein of coronavirus, they generate these memory cells that respond much faster. So instead of the virus having 2 weeks to replicate in your lungs without anything fighting against it, the fight begins within a day or two. And so those infections, if you have these memory cells, should be much less severe. And if you have these antibodies, hopefully never happen in the first place. So that is how immunity is generated. But like I said, since naive cells take a long time to be activated, you have this sort of horrifying viral replication happening for a week or 2 weeks while they're getting activated. So this is a great way to activate your immune system in theory, because look, there's tons of viral protein here. And so there's lots of things for this, for your immune system to recognize, for those naive B and T cells to find, to become activated. But it's dangerous because, the virus may spread too much. Even if you don't die, you may have lingering symptoms or experience a lot of discomfort. The beauty then of a vaccine is that it eliminates the risk, but achieves the same thing. So those naive B and T cells are there, but they don't know they're useful, right? Because until last year, right, the SARS-CoV-2 virus never existed. So they weren't useful. But we can tell those cells before we're in danger, actually, you are a useful cell, and we would like you to be memory cells that are able to do things like produce antibodies and kill infected cells. And we would like you to actually start producing antibodies right now in advance, without the risk of the infection. And so this is how an mRNA vaccine works, and we'll talk a lot more about this in the rest of the presentation, which is that instead of the whole virus, you have this lipid nanoparticle that can fuse with the cell, and it delivers just this simple messenger RNA. This RNA doesn't replicate, it's not a virus, it doesn't infect you. All it does is it has the instructions to build the spike protein. And so when it arrives in the cytoplasm, the ribosomes translate, they take those instructions and they start building spike proteins. It doesn't have any of the other proteins that the virus would need to assemble itself. And so you can't actually get infected, you can't infect anyone else, and no virus is produced, but you have a lot of spike protein that can then be recognized by those naive B cells and those naive T cells that are specific for the SARS-CoV-2 spike, and they can become memory cells that can then protect you if you were to get infected in the future, or can produce, the B cells can start producing antibodies to hopefully prevent you ever getting infected at all, which I think is a really marvelous thing. And it's not unique to the mRNA vaccines, right? So this, I hope it's been clear that the way the immune system gets activated and achieves memory is by exposing those naive B and T cells, those 1 in a million cells that are specific for this spike protein of the coronavirus, exposing them to that protein. And so there are a lot of strategies to achieve that, right? So you can just get infected with the virus and those cells will get exposed to the protein. That's dangerous, right? The virus can kill people and then in the meantime can spread to others, right? But there's a lot of other ways. So you can just inject someone with an inactivated version of the whole virus that will show the spike, or an attenuated version that replicates more slowly. That's more dangerous because it's a real virus. You can just inject the protein itself and have the whole spike protein floating in the blood, or just the portion of the spike protein called the receptor binding domain that we think is most important for immunity. So you can just put the protein in. You can use another virus to carry in the RNA, because the hardest thing is getting that message, those instructions for how to build the spike protein into a cell. But this is what viruses have evolved to do. Viruses are really good at getting into our cell, even though the cell tries to keep things out, right? That's what makes viruses so dangerous. So we can use a designer virus that just carries in that information as a way to transport that information in, or in the case of these vaccines that have been so successful, we can bypass the whole need for a virus and the potential side effects that come along with it, and just put that RNA into this lipid nanoparticle and deliver it itself. So I think the purpose of this is to show that an mRNA vaccine isn't some crazy technology. It really isn't scary. It's actually, of all the vaccine approaches, I think the simplest one, because all you do, other than maybe the protein vaccines, because all you do are provide the instructions to build spike protein, and that's it. And most other vaccines hinge on the same concept, but do it in actually more complicated ways that can come along with more potential side effects and things that could go awry. But I think, like, hopefully we can be on the same page about how information flows in the cell and how the immune system gets activated, and why vaccines work. And I think what I want to talk about now is really this question of, where did these mRNA vaccines come from? Because we'd never heard of them before, and then all of a sudden they're everywhere. And they're the most successful vaccines we have, even though we've never had an mRNA vaccine in people before. And I think there are a lot of questions around how did we develop these vaccines so quickly, that out of nowhere, suddenly we have 2 very effective vaccines in less than a year, and we're giving them to 1 billion people around the world. And I think the moral of this story is that it didn't happen quickly. Actually, it was a very long and tedious journey, right? And I hope to take you through a slice of that journey. It won't be the whole thing, because this really, I mean, was the work of hundreds, thousands of scientists along the way. So it begins before this, actually, with the discovery of mRNA in the 1960s, but I wanted to highlight this experiment in 1990. So we're talking 30 years before the SARS-CoV-2 pandemic, where Wolf and colleagues showed that if you simply inject either RNA or DNA, so the information that encodes, the instructions for building a protein, into mice, you can get mice to produce that protein. So in this case, just for proof of principle, they used luciferase. which is a glowing protein like in a firefly. And they can make mice produce luciferase, and certain cells in that mouse start to glow by injecting simply a small amount, microgram doses of RNA in black or DNA in these striped bars. And you can see that as you increase the dose, you get more expression of that protein, and over time it goes away. So it's a relatively short-lived effect, but you can get these mice to produce luciferase. And so this is sort of the birth, I think, of the mRNA vaccine field, is this idea that, look, to activate our immune system, what we need to do is get the spike protein present in the body. This is a clear demonstration that you can introduce a new protein into a mammal, a mouse like a human, using simply injecting RNA or DNA, which I think is fascinating. But in 2000, so 10 years later now, and this had been noticed before, but this is a really beautiful experiment. They noticed that if you saw before, the production lasted a relatively short amount of time. With DNA, which is a long-lived, stable molecule, that if you just inject RNA or DNA into a mouse, the mice die. And so here we have in black a normal mouse, and this is a survival curve and time in hours. And if you inject, in this case, DNA, but you see the same thing with RNA, if you just inject DNA into a mouse, all of the mice will die relatively quickly if you give too high of a dose. But if you delete in that mouse this protein called toll-like receptor 9, TLR9, all of the mice survive. And this was a really fascinating discovery, because what TLR9 is it's one of those innate immune sensors that I was telling you about before, that sort of recognize generic things that look different from us, that look like a virus instead of a human. And so it makes some sense that if you just inject DNA into a mouse, you'll have DNA where it isn't supposed to be, because DNA should all be in the nucleus. That gets detected by this TLR9 and causes this really strong antiviral inflammatory response that actually kills the mice. And so it shows that there's some need, right, to mask these DNAs if we ever want to use them as a therapy, to mask the DNA and RNA to prevent our innate immune sensors from sensing it as a virus and responding in a way that actually can kill us, right? Because if we have too much inflammation, we can die. And this was actually quite simple with DNA because it's spatial. So a lot of it is just that the DNA has to get to the nucleus. Otherwise, it gets detected as foreign. And because we identified pretty early this modification of DNA, wherever you have a C and a G nucleotide together, those are modified and methylated in mammals, but not in bacteria. And so if you just synthesize DNA and you don't methylate those, your cells recognize that as foreign. And so with DNA, we figured out pretty quickly, or others in the field figured out relatively quickly how to make DNA so that it wouldn't cause this effect, to avoid this recognition by TLR9. But with RNA, it was less, it took longer to discover and was less clear. And so this brings us to, I think, the two of the heroes of this story that actually has many heroes that we won't be able to talk about everyone. But these are two researchers from Penn, where I work now. So there's my conflict of interest, but I've actually never met either of them in person. Dr. Drew Weissman and Dr. Kate Kuricko, who really have, I think, this year gotten the recognition that they deserve for pioneering work that allowed the mRNA vaccines to be possible. And it really hinged on this observation that if you simply inject RNA into a mammal, it will be rejected and cause severe toxicity, and how to get around that. And so this is one of the key initial papers by Kate Kuricko in Drew Weissman's lab, where in 2004, they show that if you inject RNA that you produce synthetically, which is what you would want to do for a drug, you need to be able to produce the instructions that you want to inject, right? Or bacterial RNA, they cause this inflammatory response that isn't caused by DNA. So why? Why does the RNA that we produce synthetically cause the same inflammation that a bacterial RNA, which is a foreign invader, would cause? And in 2005, they published their landmark paper, for which they're probably going to win a Nobel Prize. They've won a lot of the big prizes this year for this paper in 2005. So we're talking 15 years before the pandemic, was really when the groundbreaking discovery occurred, which is that they showed these are different things that cause inflammation. And here, this tallest gray bar, this is inflammation on the y-axis. You have synthetically produced RNA. And if you treat with RNase, which degrades the RNA, it goes away. So you know that the inflammation is caused by the RNA itself. If you take mammalian RNA, you don't see that effect. And if you take bacterial RNA, you do. But interestingly, the bacterial transfer RNA, which is used to actually build proteins, has a much lower effect than total bacterial RNA. And they thought that was interesting. And one thing that was known is that these transfer RNAs are actually more modified. They have nucleotides that aren't just A, C, G, U nucleotides, if you're familiar with that terminology, to build the RNA, but they use these modified nucleotides. And so they did these experiments, and I guess we don't have to walk through all of the data, but if you see here in cells that overexpress these TLR proteins, these things that recognize invading RNA and DNA, TLR3, 7, and 8, the unmodified RNA is immunogenic and causes this inflammatory response. But if you add these modifications to the RNA, that inflammatory response is reduced. And so what they discovered is that the sensing, the reason that injected RNA is so immunogenic, causes an inflammatory response that is detrimental and can kill a mouse and would ultimately be harmful to a person if you were to try to treat them with this therapy, is because synthetic mRNA was unmodified, but the mRNA in our body is modified. It has these modifications that are characteristic of a mammalian cell, of RNA produced in a mammalian cell, and that these receptors, these innate immune receptors that are inside every cell of your body, are evolved to recognize foreign unmodified RNA. So this opens up the possibility that if you produce modified RNA, you could actually potentially inject it, produce proteins without this inflammatory response. And in tandem, other labs, it's not just the Weissman and Kuricko labs that were working on this. There were other labs in 2006 that looked at other parts of the mRNA and found in black here a way to make the mRNA produce more protein so that the ribosomes would be better at using it as instructions, and that it would last longer so that it could become more potent. So there were several groups working on this question of how can we improve RNA as a potential therapy. And the reason is that there were a lot of doubts. So DNA was seen as stable, it's long-lived. So if you want to introduce a new gene, DNA can last for a longer time, and it was easier to make DNA not trigger an immune response. And so people saw RNA as unstable, which is true. If you just have RNA floating around, it gets degraded very quickly, often within minutes or hours. It doesn't cross the cell membrane by itself. Even if it does, it won't last very long, and it will trigger this inflammatory response. And so no one was excited about RNA as a drug. And actually these papers, which we look back at now and say, wow, this was groundbreaking, this is what helped produce these life-saving vaccines, actually got very little acclaim at the time. And Dr. Weissman and Dr. Kuricko were struggling for funding and really like not recognized as doing really brilliant, important science because no one was excited about RNA. But there were a few labs within that context that were still chipping away. And they continued their work, and Kate Kuricko, Andrew Weissman's lab, collaborating, further found the mechanism. They figured out that it's really this pseudouridine modification that most increases protein expression. So you have the modified pseudouridine in black and the unmodified in gray. And you can see this huge increase in the ability to express the protein, like for instance, the hypothetical spike protein, if you use pseudouridine. And that they figured out the exact mechanism of how that happens within the cell. So during these years, these 5 years after their big discovery, they were sort of working and publishing this work in relatively low impact journals. So it wasn't that people saw this as really important stuff, but they continued to chip away. And they saw that even when you use pseudouridine, you still saw a little bit of inflammation, right? Which means that the cell senses it as a foreign RNA, and it activates this antiviral response that shuts down protein production. And obviously that's bad if you want to make a vaccine, because you need the spike protein to be expressed. And so they did further work in 2011 to show that if you purify the RNA, so when you synthesize RNA, even if you use the pseudouridine, you reduce the inflammation a lot here in red compared to blue, but there's still a little bit. And what it turns out is that there are impurities of double-stranded RNA, which our RNA is single-stranded. Some viruses have double-stranded RNA that we sense as foreign. And if you purify out that double-stranded RNA byproduct from the synthesis reaction into just the single-stranded modified RNA, you can completely get rid of inflammation. And so this sets the stage now in 2011 that the RNA technology has been optimized to the point that it really doesn't cause any innate immune response, which overcomes one of the big hurdles. And yet, most people had already given up on RNA as a therapy. And so they still were among the only labs really working on this. But they continued to push ahead. And in 2012, the next year, they showed that if you take that modified, purified RNA and inject it here in red compared to the unmodified in blue, you can get expression of this protein for several days. And eventually it goes away in a mouse. So if you inject this RNA into a mouse, you can see that the protein is expressed and actually drives a biological effect. In this case, the synthesis of new red blood cells. And you can see that the unmodified RNA is unable to actually drive the production of new red blood cells, whereas the modified RNA is. And so you see the potential for an actual mRNA-based therapy, specifically using this discovery that by modifying the nucleotides in an RNA molecule, you can avoid immune sensing and allow for protein expression that can have a biological effect, which is really remarkable. I mean, to just marvel at this and think like, wow, they can actually, after 2005, 7 years later, working in relative anonymity, they're really moving forward this technology. They weren't the only ones at this point working on it, and in parallel, and I'm not going to tell this story, but it's just as interesting on its own as the development of the lipid nanoparticles, right? Because RNA itself cannot actually get inside of a cell. So you need something to carry it across the cell membrane. And instead of using a virus, part of the beauty of the mRNA vaccines is that they use these lipid nanoparticles. And so the lipid nanoparticles were developed, and you can see on the left, injecting RNA alone, you can get a little bit of antibody response in a mouse using an mRNA vaccine. But if you use the lipid nanoparticle, you get a much stronger response, so more antibodies produced. And another lab showed that in parallel in 2013. So these lipid nanoparticles provided another advance, that these labs weren't using modified RNA. And so actually their vaccines were fairly weak, but they found that independently, the lipid nanoparticle can improve the efficacy of an mRNA vaccine. And interestingly, this second paper in 2013, they weren't using modified RNA, despite the fact that it was discovered 8 years earlier by Kate Kuricko and Drew Weissman. But they did say the very last line of their paper, they noticed that their vaccine caused this inflammatory response that actually hindered the efficacy of their vaccine. So if they inhibited the immune response, they got a much more effective vaccine. And they end their paper with this quote: "The use of modified RNA nucleotides could be a feasible and potent way to enhance the efficacy of RNA vaccines, as they have been reported to release less type 1 interferon." And in fact, 2 years later, in 2015, Kate Kuricko and Drew Weissman at Penn used this lipid nanoparticle technology for the first time with their modified mRNA molecules and show a very effective and sustained production of protein in a mouse for out to 10 days after the injection, which I think is also interesting to think about for our mRNA vaccines, that the spike protein is really only there for most likely less than 2 weeks. But one thing that I just think is great about this study, which shows that you can get protein expression quickly and sustained over time, is that they have the intramuscular injection here in sort of this pink color, which is actually lays the foundation for how we develop these intramuscular vaccines that a lot of us have been receiving in the last year. And other labs continued. So Kate Kuricko actually in 2013 left Penn to join BioNTech because there was a lack of funding and interest in their research academically. But this company, BioNTech, which you may have heard of for developing the Pfizer vaccine, wanted to use mRNA technology to produce new therapeutics. And so she actually left Penn to join them. But other labs carried on the mantle, and you can see that actually it started to pick up steam. And in 2015, you have a paper that finds a new modification that is even better than pseudouridine. And they have this quote that I think captures something really interesting that says, messenger RNA as a therapeutic modality is becoming increasingly popular in the field of gene therapy. The realization that nucleobase modifications can greatly enhance the properties of mRNA by reducing the immunogenicity and increasing the stability of the RNA molecule, the Kuricko paradigm, has been pivotal for this revolution. But it took 10 years for other people to recognize that, which I think is a fascinating part of the story. And then it really pre-culminates, because it doesn't culminate until COVID-19, with this demonstration that a Zika virus vaccine, Zika virus mRNA vaccine in 2017 by Drew Weissman's group, it can be extremely effective in mice. So this is the vaccine activating or generating antibodies. And if you then challenge those mice with the virus in gray, unvaccinated mice, they get infected and you can detect the virus. Vaccinated mice never get infected despite receiving the same challenge. And you can do the same thing in macaques, in non-human primates, which are very similar to humans. And you can give them an mRNA vaccine. They generate these neutralizing antibodies after the vaccine. And if you expose them to virus in black, they get infected if they were not vaccinated, but the vaccinated animals are not infected. So this got published in Nature, which is the highest of high profile journals. So now all of a sudden in 2017, 12 years after the real breakthrough, mRNA vaccine technology was becoming extremely popular, and optimism about it was starting to grow. And so those of us in the field were not so surprised that mRNA vaccines ended up being some of the leading candidates when SARS-CoV-2 happened, because this had been developing in the few years prior. And then, of course, SARS-CoV-2 happens. So on December 31st, 2019, the World Health Organization is informed about the pneumonia cases in Wuhan. 8 days later, on January 7th, the virus is isolated and sequenced for the first time in China. And 3 days later, that sequence, the sequence of the viral genome, is made public. And so this was the moment that the world realized that the cause of these pneumonia cases was a coronavirus that was highly related to the original SARS coronavirus. And only 3 days later, both Pfizer and Moderna had already finalized their vaccine formulations, which is remarkably fast, obviously. And here in August 2020, we got the first results. This was a very happy day in our lab. You got to see the first, we had nothing to do with developing the vaccine, but for the world, we got the first evidence that the Pfizer vaccine would work. And this was before we knew the results of the clinical trial, but they showed that this mRNA vaccine was generating really good levels of antibodies against the SARS-CoV-2 spike protein. And then in December, both Pfizer and Moderna released their clinical trials. And I almost get emotional when I look at the graphs, because after all the suffering and death of 2020, that at the end in December, we saw like some of the most beautiful graphs I've ever seen, which are just so clear, the placebo versus the vaccine in terms of the accumulation of infections and how remarkably effective these mRNA vaccines were. And here's a photo of Kate Kuricko and Drew Weissman, who really pioneered the technology, receiving their vaccines at Penn, which I think was a beautiful and historic moment. And so I just want to highlight really quickly before we run out of time, this question: how could these vaccines be formulated so quickly? And because it really only took them 3 days from getting the sequence to finalizing the vaccine. And then it took 12 months to go through testing to make sure they were safe and effective before we could start administering them to the population. So how could they possibly design a vaccine in 3 days? It's because of the work of other scientists who weren't lauded for what they did. They weren't famous, they weren't highly regarded, but they did science that they thought was interesting. And so after the SARS scare, which didn't become a pandemic, but easily could have, in 2004, this lab, Peter Collins, did a study to show which parts of the SARS coronavirus were important for generating protective immunity. And this is actually where they found that it's really only the spike protein that matters. So as soon as the SARS-CoV-2 sequence was released, all of the focus was on the spike protein, because we already knew from 2004 that a related virus, the spike was the protein you wanted to target with the immune system. There had also been phase 1 vaccine trials. These vaccines never went further than phase 1 because SARS was no longer infecting people. But there were phase 1 vaccine trials showing that with a 2-dose regimen, you could generate strong neutralizing antibodies against the original SARS. This may look familiar. We use a 2-dose regimen for the SARS-CoV-2 vaccines. And another phase 1 trial using only the spike protein, showing that just immunizing with the spike also generates good neutralizing antibodies. And then a very important breakthrough in 2017. Again, nobody really cared about this because SARS and MERS were not pandemic viruses. But this lab, the study led by Paulson and colleagues, in 2017 showed that you could introduce 2 small mutations into the spike protein to stabilize it. So what does that mean? If you look here at these really beautiful microscopy images, you can see kind of 2 different versions of the spike protein for both MERS and SARS. And this blue version is the version of spike that you actually need antibodies to recognize. But a lot of the time, spike takes this other conformation that can distract your immune system and cause you to generate antibodies that are not so useful against the virus. But they show that by introducing 2 small mutations and using this, you can stabilize the spike protein so it only takes the desired conformation. And if you immunize with the stabilized spike protein, you get a higher neutralizing antibody response. So this happened in 2017. You know, you can kind of shrug, never think about it again. But then when Pfizer and Moderna were designing their vaccines, they were able to introduce those same 2 mutations to stabilize the spike and produce a highly effective vaccine. And actually other vaccine candidates that didn't use the stabilized spike from this paper were not effective. So it actually was a critical breakthrough for the success of the vaccines. And again, happened in relative anonymity years before the pandemic ever happened. And then lastly, I want to talk about how the vaccines could be tested and approved so quickly, because that's a big question people have, because normally to develop a vaccine takes 5 to 15 years. You do a phase 1 trial, a phase 2 trial, a phase 3 trial, seek regulatory approval, and then begin manufacturing, all sequentially. And the reason is that these are extremely expensive. And so the mRNA vaccines, just to make a small point, were not developed faster because they skipped any of these phases and didn't complete clinical trials and didn't test for safety, but rather that the companies took on the risk of beginning a very expensive phase 2, phase 3, and phase 3 trial before they knew the results of the earlier trial, which carries enormous financial risk. But because of the skewed financial incentives during the pandemic, a lot of companies chose to do this and actually begin manufacturing before they knew if their vaccine would ever work and before it ever received approval, which several companies whose vaccines are still not approved have lost an enormous amount of money. Obviously, for the ones that succeeded, they will make an enormous amount of money. So there's financial implications in all of this. But I think it's really these skewed financial incentives that drove the speed at which these vaccines were able to move through trials. And then lastly, that it's not just that every vaccine worked. So this is from September of 2020, it's a summary of all of the vaccines that were being developed. So each circle represents a different vaccine of a different class. So the RNA vaccines are these in light blue. And in September, they were in the Pfizer and Moderna were in these phase 3 trials. But all of these other vaccines that you've never heard of either failed or never made it through the trials to date. So just to say that it's not that making a vaccine is easy, or that the first thing we tried worked, but rather it took the effort of a lot of people just to find a couple of successful vaccines. And so that is really, I think, like hopefully like the fascinating, beautiful history that shows that to make a successful vaccine is a long journey, going back 50 years, 70 years, to 60 years to 1960. I sort of walked you through this left side, which is the development of mRNA, but just to say that in parallel, developing the lipid nanoparticles and putting them together was just as essential. And many discoveries along the way ultimately led to the success of these vaccines. And I just want to end this section with a few quotes from the people involved. So Kate Kuricko said, everyone just incrementally added something, including me. So she's getting a lot of praise, deservedly so, for persevering in this work when no one else believed in it. But she also has the recognition that this required thousands of scientists doing their part. And then Peter Cullis, who's credited with pioneering the LNPs, said, you really can't claim credit. We're talking hundreds, probably thousands of people who have been working together to make these LNP systems so that they're actually ready for prime time. And that has been my experience as well. So just very quickly, this is the team of scientists I work with. So I came to join John Ware's lab at the University of Pennsylvania, specifically to study immune responses to COVID-19. And I work with Divij, Socrates, Rishi, and Ali as co-leading and trying to make important discoveries about the vaccine, which has been really interesting. And I don't think I have time to talk about our research, which is okay. It's much less interesting than the research people did before. But we've just shown that, for instance, early on in the vaccination schedule, we were able to show that people who had recovered from a previous infection really only need one dose of the vaccine, whereas people who hadn't recovered really need both doses to achieve good immunity, and that the vaccine generates better immunity than infection alone. Those were two really important discoveries that I think we were able to contribute to, along with a dozen other labs around the world, to try to understand how to use these vaccines well and how to make sure people really have robust immunity and protection against these viruses. And then we've done a more thorough study looking at all of those aspects of the immune system that I talked about at the beginning: antibodies, memory B cells, memory T cells, and how they respond to this vaccine. And the high level view is just to say that the vaccines generate really beautiful, strong memory cells that look like they'll help you be protected for a long time. And we can talk much more about the immunology if you have questions, or if you want to contact me afterwards even. I'm happy to talk about all of the things we've done, because really I don't study the development of these vaccines, I study these immune responses, but it's, I think, really fascinating to look at the story. And so I just want to close with a few comments on what have we learned and where do we go from here. So I think, what have we gained? For sure, many of us have gained immunity, hopefully from the vaccines, or God willing, from surviving a coronavirus infection. A lot of us have gained immunity, but I think we've gained a lot more than that as well. So I'm going to stop sharing my screen and just talk to you. So, I mean, what have we learned? My experience, I don't know, to do science during the pandemic was really an interesting experience, because immediately, like, this new virus arrives in late 2019, early 2020. I was a virologist at the time studying HIV. There's this huge desire to know, like I cared about HIV. It kills 1 million people a year around the world. And immediately it's like, this new virus, like, we need to know it. We need to know it. And there's this desire almost to try to save the world, because you see all the people dying and you're a virologist, right? And so how can I help? And immediately, like, we start thinking, we're a virology lab, how can we study this virus? And immediately, every project you think of, every set of experiments, I could see right away the disproportion between what I could do and the needs, right? And the desires that we had, the things we needed to know and what the world actually needed, and my little experiments that I could do. And it becomes evident right away that like the science you do isn't going to save the world. And actually, even now in hindsight, some people did develop incredible mRNA vaccines that save lives, and yet I think we can look around and say like they haven't saved the world. You know, the pandemic is still happening, and all of our human needs remain in play. And so, why do we do it, right? Because in the end, like I have totally shifted my research focus toward COVID-19, even though I know that I'm not going to save the world by what I do. And I think what I've really learned, if we've learned anything about biomedical science, is that it brings us back to the fact that we do it. I do it because I'm curious, because I really want to know, because like this new virus is interesting to me, and because I'm desperate to know how the immune system is activated by this virus and how it's activated by these vaccines, and whether the vaccines are going to work, not just to save the world and for policy decisions, but because I want to know, the whole world wants to know. And so it's been really, I would say the first thing is that it's humbling to be studying something that everyone wants to know, which is for sure not the experience of a scientist. Usually no one cares about what you do. And also came with, I think, the thrill of, you know, those moments where, you know, we're doing an experiment and we see something about how the vaccine works. And for a moment, you're the only one in the world who knows, or one of a few people in a few labs who might know, who have that data. Which immediately like brings out the need to share it, and to share it as fast as you can. But first of all, to make sure it's true. And I think that was an interesting thing that Giulietta brought up at the beginning, the sort of relationship between making sure what you've discovered is true, or as close as you can be to being sure, while also sharing it quickly, was one of the dramas of the pandemic. And then the second is that doing things as a team can be extremely productive. So because we wanted to know so badly, we all work together, and scientists from different institutions, all of Penn, basically worked together on a single vaccine study, leveraging the skills of different labs and different people within those labs. And labs from around the world shared resources with each other. All of my work was totally dependent on a group from San Diego sharing one of their resources. And this allowed, with this team science, like if we all agree on what we're interested in and we put our egos aside and do it for love of the science and really like the need to know, instead of our own ego or our own personal glory, I think we were able to achieve much more, and to work harder because we were working together. And I think that probably a lot of the physicians here had the same experience, that during the pandemic, we worked harder than we've ever worked as scientists. I've never worked this many hours and this hard. And yet it was hard, but it was tireless. Like I wasn't weary. And it was because, I really think it was because I was doing it with my friends, like together, because we all shared, it was motivated by this real need to know the answers and the collective pursuit of them, that like being there with my lab mates every day made it possible. And then the last thing I really want to say is that I think in Drew Weissman and Kate Kuricko, we have great examples of what science is and how it works, that the heroes of the story are normal people. They aren't amazing because they were superheroes or mad geniuses. They were just people who loved their science. And like Kate Kuricko is just an mRNA nerd. Like if you hear her talk, like she just loves mRNA. And didn't want to give up on it, even though everyone else did. And so they didn't do it for fame. They weren't famous. They weren't even getting grant money or to save anyone's life, but simply to keep discovering something. And because each discovery opens up more questions. And so all this time they did this work, little recognition, little funding, but for the sake of the science itself. And so we look back and we see a long journey, not of like people who thought, I'm going to make a vaccine to save the world or to save millions of lives, but thought, I'm interested in why RNA activates the immune system. And that each step along the way for those 50 years of developing the technology, but especially the last 20, is done for the love of that one tiny aspect of reality, of like what a pseudouridine does when it's put into an RNA. which is done for its own sake, not to save the world, but because it's interesting and it's beautiful. And the fact that people do it, slowly you can see that like, this is the way that we build something. And so what does this teach me, at least I think, about biomedical science? I think it shows that it works best when we pursue what is interesting for its own sake, rather than seeking immediate cures. Because if you're looking immediately to cure something today, you can only use the tools you have available today. And yet, a lot of time what's needed is a new tool. And the mRNA vaccine was a new tool we didn't have until 2017, when that Zika virus trial started. And it took 30 years, it took decades to develop, right? But because people invested in doing that for its own sake, not for glory, not to save lives today, but because it was interesting, they developed a new tool that ultimately could save lives. And so I think having this perspective, that what we desire is the fruit of a long journey and not a short-term attempt. And then I think the last thing that we really learned, that I learned personally, is that science needs to be done with humility, because we never know the whole truth. And so, especially early in the pandemic, but every day as a scientist, you want to draw conclusions, and you should, from the best available data, and try to build frameworks and understanding and make policy decisions based on the best data that's available. But we have to have humility and be aware that we could be wrong, because we never have the whole truth. The world is a mystery. The reason we do science is to uncover tiny aspects of that mystery, and reveal them. But we never see the whole, and we never will. And so we need to approach everything we see with humility. And first of all, it was my own experience that like early on in our vaccine studies, I thought I was even like making sarcastic memes, sending around our lab. Like I thought there's no way we're ever going to need booster vaccines. These vaccines are so good. And then 3 months later with my own data, I was like, oh shoot, actually, we probably will want to give people booster vaccines. And you know, early on, I thought that the immunity would be better. It looked like the immunity was better in people who had COVID and then got vaccinated than people who just got vaccinated. But actually now, 6 months later, out from those first vaccines, we see that actually they kind of end up at the same place. And so like things that I thought were clear interpretations from our data actually have turned out not to be real. And I think that it shows the need to be humble and acknowledge what we don't know. And I think like in hindsight, it's also beautiful to say like, we never know the whole truth. And that's what makes science keep going and so interesting. Because imagine if Drew Weissman and Kate Kuricko had stopped in 2005, right? That was the big discovery. That's the discovery that's going to win a Nobel Prize, hopefully, because they deserve it. But if they had stopped there, those other 15 slides I showed you would have never happened. If they were happy thinking, wow, we've finished it, we discovered it, we would never have an mRNA vaccine. And so the fact that we're humble, that we recognize that we don't know the whole, actually is what makes science interesting, that every discovery urges you onward, opens you up to new curiosities, and new things you want to know. Like, I don't know, like if the person you love tells you something about themselves, you don't just, oh, that's nice. You have a million more questions about it, right? Because now you know, like, you know, some new thing, it opens up 100 new, even more curiosity. And I think to feed into that is really important for science and for the development of these vaccines. So I went long, I'm very sorry, but I hope it was useful.
Thank you, Mark. Thank you. Really, thank you for, because it was really fascinating to hear you and all these stories. We receive a lot of questions. Many people send a lot of very interesting and good questions. Unfortunately, we are running a little bit over time. So we would like to ask everyone who sent questions to send them to the MedConference email that you can find on the slide that we are putting on in the break times. And we will collect all these questions and we will send them to Mark
Julieta, can I, and so I saw like 10 of them are all about third doses.
I know, yes, yes. If you want briefly reply to that, that would be great. Yes.
So basically you have antibodies that protect you from ever getting infected in the first place. And then you have memory B cells and memory T cells that can, if you do get infected, respond much quicker than they would have the first time around, right? So what we're seeing with these vaccines is that your antibodies go really high immediately after the vaccine, which is why you get those numbers like 95% efficacy from the clinical trials against infection. But over time, those antibodies decline, and they gradually decline. And as they decline, it's not that one day you're immune, the next day you're susceptible, but you become gradually more susceptible to getting infected over time. Your memory cells, your memory B cells and memory T cells are rock solid. They look really good. They look like they might last your whole life. You never know, but they aren't going anywhere. And so as the antibodies decline, people will become susceptible to infection. Those infections on average, because of those memory cells, should be, and the data right now show that they are, less severe than getting infected without having been vaccinated before, because those memory cells are able to respond in 1 or 2 days instead of a week or 2 weeks. So you start fighting the infection sooner and you're less likely to spread it to other people, but you may still become infected. So that's the reason probably for the increase in breakthrough infections over time. And also the reason to get a third dose is not to help those memory cells, but it's to temporarily jack your antibodies back up to a level that protects you from infection. So it sort of sets up a scenario where if you get 2 doses once in your life, you're probably at least prepared to have a less likely chance of getting severe infection for the rest of your life. But you'll become susceptible to infection over time as the antibodies decline. If you keep getting a booster, you can keep those antibody levels high enough that you never get infected at all. So that's a policy decision people have to make, and also a personal decision about how to approach that. But.
Thank you. Thank you so much. That was really needed, actually, and really clear. So thank you again for this really fascinating journey and for sharing with us all the backstage of everything that was going on in the research field, that is still going on in the research field. And the way that you presented it really made me think that we are really standing on shoulders of giants, even when we don't realize that. So again, if anyone has any other question, please feel free to send it through email, and we will make sure to forward them to Mark and to share his answer with you.
Yes, thank you. I'm happy to answer any questions. So.